At NHBS, our purpose is to support those who are passionate about wildlife, ecology, and conservation. We stock a variety of books and equipment to suit the needs of marine conservation professionals, and our innovation and research team are here to help develop custom products for any project. We are happy to provide advice and to support you before, during, and after your purchase. Visit nhbs.com today to find out more.
Good morning. Welcome to the Assemble Plus conference and this second session this morning on cryopreservation. And to lead this meeting is Stefania Paredes from University of Vigo. Please, Stefania, you can carry on from now.
Thank you, Adelino. So first of all, I would like to welcome you all to the session. After this nice video and the introduction from Adelino, we are here kind of gathered to discuss about cryopreservation, and we have people joining us from researchers from academia, researchers from private companies, and we also have companies and technology providers. So thank you all for gathering here and being willing to talk about cryopreservation. Two little things about housekeeping: please keep your microphones off. And any questions to the speakers can be written down on the chat, and we will address them on time. So we will have today Neil Stockham, Sofia Blanco, and Juan Asturiano here with us. And they will be giving a little presentation about their work. And we will be getting one or two questions, but then afterwards will be a round table at the end where we will answer all the questions and discuss a little bit all together. So without further ado, I would like to introduce Neil Stockham. He is from Planktonic. He's the CTO of Planktonic, which is a Norwegian company dedicated to the development of live feed and preserved feed for aquaculture. And they have developed a unique approach by cryopreservation of crustacean nauplii for aquaculture feed in the future. So I'll just leave the floor to Niels so he can show us what they're doing in Planktonic.
Yeah, thanks a lot, Stefania, for letting me have the possibility to talk about our cryopreserved live feed for aquaculture species. So my background is within marine food webs, planktonic food webs, and not cryobiology. So I would say I'm self-educated in this field. So in aquaculture, the marine larvae, they require live prey the first days after hatching. They need a prey that has some movement. And it's used a lot of rotifers and artemia annually. And this type of live feed, they are suboptimal for marine larvae in aquaculture. You will experience low survival, low growth, and low quality larvae. So we, Planktonic, we offer a natural live diet for marine larvae. So that consists of barnacles. And we use two species, Semibalanus balanoides, which is intertidal species. They can replace rotifers and artemia. Balanus crenatus, a subtidal species, can replace rotifers. And we are using, as you see here, the stage 2 this diagram, stage 2 nuclei. This is a natural live feed for fish larvae, which they are adapted to eat, and therefore we think it's optimal feed for marine larvae. And they meet the nutritional requirements of juveniles in aquaculture, as we see on growth and survival and quality of fish larvae. And we are in revenue of this type of feed. We sell now this cryoplankton, which we call it, to up to 20 different hatcheries, which are producing kingfish, ballan wrasse, lumpfish, and other species. So we are cryopreserving in industrial scale. We are adding a cryoprotectant agent. We are using a controlled rate freezer, which can hold up to 600 kilo for each cycle. And we have about 300 divars, each of them 175 liters, that can hold 24 tons of cryoplankton. So when we started to cryopreserve those barnacle offspring, we had the feedback from experts in the field that too high concentration of CPA that would lead to high or full mortality at equilibrium. Long freezing protocols would often be lethal to the organism. If you combine high concentration of CPA and long freezing, that will also be very lethal. And it's difficult when the organism is too large. And suitable volumes was not in industrial scale. They were in straw or vials. But now we are using high concentration of CPI. We have been using DMSO, ethylene glycol, propylene glycol, all in very high concentration, and ethylene glycol more than 7 molar. Freezing take longer than 8 hours, and we combine this high concentration of CPA and long freezing, and we use an organism that is more than 300 micron. And we are cryopreserving by tons, not straw or vials. And yeah, we are aiming to freeze up to 20 tons this year. So with this protocol that differs a lot from other protocols, it was quite easy to obtain a patent. So now we have a patent in several countries, and more is to come. Planktonic, we have 5 aquaculture licenses for producing barnacles in extensive systems. And each locality have the capacity of producing 100 ton, so combined about 500 ton, and more to come, both in Norway and abroad. And we see that we could soon replace all rotifers and artemia in aquaculture in a few years with this method. So using the cryoplankton, we have very strong, unique selling points for this cryoplankton compared to the conventional live feeds on the market today. First of all, it's easy to use. When you use conventional live feeds, it's often up to 50% of the resources at the hatchery that are used for producing live feed. Here it takes maybe 30 minutes or so per day to thaw and to wash and to revitalize. And it's unsurpassed biosecurity on this cryoplankton compared to other type of live diets on the market. And that's because it's eggs that are extracted from the adult barnacles, and eggs, they are in principle sterile. And they have not opened any mold or anything that they could eat something that could be bad for the fish or for the aquaculture. And the hatchery managers, they have asked us to analyze for pathogens, and they have been given us a list of pathogens that we are analyzing And now we have analyzed more than 200 batches and found no of those pathogens we have analyzed for. And the freezing protocol itself kills a lot of microflora. With the high CPA used, that's lethal to many bacteria. So it's a very low amount of bacteria in the end here. And we see that it's an increased growth at the hatcheries, especially for sea bream and sea bass and kingfish. For kingfish, it's we have reports that they are growing the fish 40% faster up to slaughter weight, just by introducing a live feed for 20 days or so in the start. And we see that the fish get more robust. We see increased stress resistance of the fish. That means the hatchery people can handle the fish without much loss. And then they have a higher survival in the end. And this type of product is more reliable in delivery compared to other type of live feeds. Because you do not have to be dependent of a live feed culture. You have storage of cryoplankton in liquid nitrogen that have an infinite shelf life. So if you combine all these arguments, then you have a very low cost per produced larvae at the hatchery. So here are some pictures from a large hatchery that are using our cryoplankton on a regular basis. They have replaced all Artemia, and now they are about to replace all rotifers as well. They are the largest customer. I think they buy 3-4 tons per year of the cryoplankton. And here you see they are taking out of the divars pelleted cryoplankton that are soaked directly into the liquid nitrogen. No plastic wrapping or anything. It's free in the liquid nitrogen. And here you see the person takes a bucket with the cryoplankton, tow it in seawater, rinse it and filter it, filter out the CPA, transfer it to a milk chiller, where they start to revitalize the plankton, and then feed to the fish tanks. They have 16 tanks of 25 cubic each, and yeah, they have a far higher survival with this diet compared to rotifers and Artemia. It's a huge improvement. So now I think I used my 10 minutes. So thanks for your attention.
Thank you, Niels. This is super interesting. I'm really impressed by the huge scale of all the production. So if anybody has a quick question, we can actually do this now. Otherwise, we can wait till the end. But meanwhile, so if I understood correctly, is the plankton pelleted inside of the tanks?
Yeah.
Without any wrapping or, you know, bios, bags or anything?
No, yeah, that's right. That's correct.
Wow. Wow, wow, wow.
So that makes it much easier. We started with plastic wrapping, but that was a lot of work for the hatchery people to remove, and so it's much better when it's free like this in the liquid nitrogen, so they can take a scoop and take everything out.
Yeah, I was going to say, how much does it fit inside of each one of the doers?
It's 80 kilo in one doer, and then you have close to 100 liter of liquid nitrogen. So holding time, yeah, two months or so before need of refill.
Okay, so Christine Campbell has a question, and she wants to know if you have to feed the naupli at any stage, or just directly for the fish?
No, we don't feed any of the naupli. are freezed as eggs, and they hatch when they thaw. Or it could be a combination of a first stage nauplius, which are a non-feeding stage, and eggs in a mix. But they hatch all of them after cryopreservation.
Okay.
So it's no need of feeding algae or enrichment or anything. It's just pure diet like this.
Okay, thanks. So we will just move on to Sofia Blanco. I think she's here with us as well.
Yes, hi, can you hear me? Well,
yeah, we can hear you.
Okay, great.
Can you share your screen with us?
Yes, it's the first time I do this, so be considered with me, I think. Can you see?
Sofia is a young researcher from the University of Vigo, and she's here today representing the project Meet Unisex that uses, among other different tools, uses cryopreservation. And this project has recently been awarded Sherpa Domar Awards on Entrepreneurship. And well, I'm just gonna let Sofia tell you all about it.
Okay, can you see my presentation? The first one?
Perfect.
I'm moving, okay, perfect. So thank you very much, nice to be here. And thank you, Estefania, for giving me the opportunity to present our project and our work based, as you have said, on the production of unisex lines of Galician mussels, Mytilus galloprovincialis, and its relevance for the aquaculture and the canning sector, and also the crucial role of the cryopreservation in this process. Okay, can you see past the Okay, perfect. So we have been observing a number of requirements for the optimization and introduction of molecular techniques to improve mussel production, such as the preference for orange-colored and good-sized individuals, the need to increase the value of mussels and its derivatives, a product whose production has remained static over the years, and finally, the need to be able to access to mussel seed all over the year at any time without depending on the spawning season. So in response to these needs, we propose the development and production of unisex lines of female mussels, large and orange, the use of the oocytes for caviar production, and the cryopreservation of those unisex lines to make them available at any time. These proposals, these answers, are based on our main line of study in our group, the study of the double uniparental inheritance. So I'm going to try to be clear in this part to make it easier. So this mitochondrial inheritance is unusual, but has been described in more of than 100 of bivalve mussels. And it's different from any other type of mitochondrial inheritance that we are used. It's different from the mammals or even the humans, because here we can describe two types of mitochondrial DNA lineage, the female or F type and the male or M type. And here in this type of mitochondrial inheritance, the M type passed to the next generations. It's different from humans when the sperm mitochondria get removed or deleted from the embryo. In this case, we can see, as you can see in the picture in letter A, how those 5 blue spots representing the sperm mitochondria get in a dispersed pattern or moving randomly in the embryo in the first stage of development in 2-4 cells. Whereas in the letter B, by the other hand, we can see a different pattern where the blue spots, the 5 sperm mitochondria, get aggregate, not dispersed, aggregate group next to the phorocleavage. In the first case, in letter A, the embryo will develop as a female, whereas in the letter B, the embryo will develop as a male. So here we can clearly see a correlation between the mitochondrial inheritance and the sex of the individual, the future embryo. Also, we know that these movements, these patterns of the mitochondria are controlled by the female. So the sex of the future embryo is going to be controlled by the mother. Add to this, we also know that there are mothers of mussels that produce mainly female embryos. So those one are interesting because those mothers are going to produce those unisex female lines that we want to select and cryopreservate. So knowing this, we can label the sperm mitochondria with MitoTracker green fluorescent marker and then follow their movements, their patterns into the embryo. By this way, we can select those mothers that produce mainly females because we can know the sex of the individual before they're developed into an adult. So this is very useful. This technique has been seen by my tutor Angel Perez at the Dr. Kensington laboratory in Canada with Mytilus and the objective of this project is to repeat this technique, this way to work, but in the TMAT with the collaboration of the group and of course with Estefania, in order to obtain those lines, unisex lines, but with the Galician mussel Mytilus galloprovincialis. So here is where cryopreservation is a key point because we want to conserve those unisex female lines over the year, all the year without depending on the cycle, season of spanning of the mussels. So the main idea is to use liquid nitrogen as a storage medium and ethylene glycol as a cryoprotective agent in order to cryopreservate the larva stadium. Here we need the collaboration, of course, of Estefania to optimize and to really know what's going to be the protocol to follow in order to later, the key point is to reactivate the metabolisms of these larvae in order to get them viable any time of the year. So the cryopreservation is to conserve the larva, the unisex line over the year. Also, on the positive side, in terms of funding and support, we have obtained a project from the ministry, the mitogenomics and mitoproteomics of marine mussels with double uniparental inheritance, in which Estefania also participates. Also, our project, Meet Unisex, has been first selected in the SERPA Domar project or SERPA Journeys. This program is more focused into the business way, into help us for project our idea of the unisex mussel lines. Finally, we also have the support of the entire team and the supervision of international partners and collaborators in Greece, Seattle, and Swansea, all of them researchers of great renown and prestige. Thank you very much for your attention. Thank you. Okay.
Thank you, Sophia. If anybody has a question, we can relay it right now. I think it's very interesting that we can actually preserve these lines. This can also apply to any other kind of mollusks. We've been working on mussel cryopreservation for a while, and now we can guarantee that we can preserve those lines. Although we don't have, means to work in, the amount and quantities that Niels just showed us before, but still we can cryopreserve a huge amount of mussel embryos and larvae to work with. So yeah, I think it's going to be a great collaboration so far. Without further ado, we'll just move to our next presenter,
Okay.
Juan Asturiano. Juan, are you there?
Hi, good morning.
Good. So
let's check. Okay.
Can you share your screen?
Yes, let's go.
Okay, Juan is a researcher from the Animal Science Department, the Research Group of Aquaculture and Biodiversity at the Polytechnic University of Valencia. And he's been involved in several actions regarding cryopreservation. Aquagamete, for example, many of you probably know the project. And he's also been working on really nice projects with eel conservation. So some of his research is done, I guess, in collaboration with the Aquarium of Valencia, but well, Juan will tell you all about it.
Okay, good morning, Estefania. Thank you for the invitation, and good morning, all the rest of our colleagues. I would like to say thank you for your nice invitation and to give a very brief overview of our recent challenges and our recent results, at least mentioning some projects, including some cryopreservation techniques. In fact, I want to tell you about three projects. One is called Ilgonia. It's funded by the Spanish Ministry of Science and is dedicated to eel spermatogonia. The second one is dedicated to lasmobranchs, both different species of sharks and rays. And the third one is centered in some local species of little piprinids that are basically autochthonous species in the Valencias area. The Ilgonia project tried to get the surrogate production of European eel gametes. That means to make that other species like terrafish or European sea bass or common carp produce the sperm of the eggs of the European eel. To do this, the idea is getting the spermatogonia from adult European eels, and then when we isolate this spermatogonia, we have the possibility of storing them through the cryopreservation techniques and cryobanking, or directly go to inject this spermatogonia in the larvae of these other species that are the animals who are going to carry this spermatogonia until producing these gametes at the end of the process. This is the idea. For the moment, we have developed the protocols to isolate the spermatogonia. We have characterized these cells and we have measured the level of expression of different spermatogonia biomarkers, the genes, and we have developed the techniques for cryopreserve and vitrify this spermatogonia. Concerning the lasmobranch projects, the idea is that we know very little about many of these species, and the idea is trying to know more about the life cycles of these animals, trying to compare the reproductive anatomy of this animal. This is quite different, surprisingly different between one species and the others. To know, for instance, how to cannulate them to get the sperm in live or in post-mortem animals. We have learned a lot about how to handle and how to evaluate the sperm of these animals. And of course, we have been working with the cryopreservation protocols for this species. And the idea is trying to use these relatively small species that they are part of the bycatch that arrive to our ports in Spain to develop these cryopreservation techniques to be applied later in the future in big endangered species. We have collaborated, as you said, with Oceanographic. This is the aquarium that we have in the city of Valencia, and with these facilities here, we have the chance time to time to work with big species like this North shark. And very recently, for instance, at the beginning of the month, we have the chance of working with some samples from this blue shark who arrived to the coast, dead here in near Valencia, and we took some samples from these animals, and we have been working with these samples. For the moment, we know better what's the morphology of the spermatozoa of this species that is critically endangered in the Mediterranean, and we have developed, or we have tested the protocols that we have prepared, we have fixed with this model species, this little model species before, and now we know that we can apply these protocols in these animals, in these critical endangered species, and cryopreserve the sperm of this blue shark. Regarding the morphology, well, we are learning a lot about the morphology of these cells in the different species, even well, finding some similarities with birds' spermatozoa. I have no time to giving you details on all this. And for the moment, we have been working with 9 elasmobranch species. 4 of them in the left are endangered species, and two of them, the blue shark and the bull ray, are considered critically endangered species, at least at the Mediterranean Sea. So for the moment, we have applied our protocols to this fish, and we are trying to publish the first protocols for the cryopreservation of the sperm of all these plasmorganic species. And finally, talking about this cryoval project, this is dedicated mainly to this little fish, what we call Partet and Samaru. They are called Aphanius iberus and Valencia hispanica, and they are fish that have very isolated populations living in freshwater spots near the coast. So we have always the risk of losing biodiversity or losing population of these animals with fire or with a problem of pollution or something like that. So we are trying to collaborate with the local government, who is working many years ago trying to preserve this species, and arriving to or giving one innovative approach to these programs from the cryopreservation point of view. So this month, very recently, we have published the first protocols for the cryopreservation of these animals, and as you can see, well, the first trial have got some survival of sperm cryopreservation of these species in both cases. We have started as well a preliminary study of the morphology of these cells, of this spermatozoa. And well, here you can see the faces of the people involved in the project. Basically, Victor Gallego, who is one of those of the group, Pablo Garcia, who is a PhD student, or myself. And if you want to see more details or, well, giving a better idea of the things we do, you can have a look here on our blog or just contact me. Thank you very much.
Thank you, Juan. Does anybody have any question about the work that Juan and his team are doing? We have a couple of minutes to do that now. I would guess it's quite difficult to try to develop a cryopreservation protocol for animals that are so, I mean, they're not commonly around. So you have either to count with the collaboration of an aquarium or hope that they will just, you probably go sampling and tag them for a little bit and take a sample, or you work with animals that have been washed out in the beach.
Yeah, it's a bit tricky. That's why we have decided to plan our research in this way, trying to work with animals who come with the fisherman every single day, and the fishermen are very we have a very good help from these guys, and we get all the animals we need. And the idea has been to develop the techniques with these samples from these easy species, and then trying to apply these techniques whenever we have the possibility, whenever we have the chance, from these animals arriving to the coast or using the aquaria specimen.
Thank you. Well, if there's no questions right now, I will just show you my presentation and just let's see. Okay. And I'm going to be showing you some of the things we have done with within the project Assemble Plus. So one of the main ideas is that within the big purpose of a project as big as Assemble Plus, we could gather a group of people interested in developing cryopreservation, improving techniques, just changing ideas. So as you can see, Assemble Plus is big, and some of us had an interest in developing cryopreservation, had the equipment, and had the idea on how to apply and use cryopreserved cells to different purposes, from research to aquaculture. So we've been working for almost 4 years now. And we finally published one of the deliverables of the project was a sample of protocols for people to use and try in a controlled manner and very well explained step by step, so anybody could actually use them, either in academia for the research purposes or in a private company. So some of them partners we have come from CCMAR in Portugal. As you can see, they have great equipment. This is the lab of Elsa Cabrita in Portugal. And they have portable freezers, bench biofreezers, and a huge team of tools to assay post-thaw quality of the cells they're cryopreserving. And then at the end, I will show you some of the protocols we've developed together within Assemble Plus. From CCMAR to the University of the Basque Country to Plancia Marine Station, when they have environmental biospecimen bank, where they cryopreserve cells from fish, from marine mammals, from mollusks and sharks. And they have them on this great facility, this biobank. So if you want to work with the samples, they do have the opportunity of sharing the samples or the information with researchers. Well, this is my lab in the University of Vigo. We also have different kind of freezers, a very small biobank at the Ethimat Marine Station in the Centro de Investigación Marina. One of the equipments that is quite unusual is we have a laser to thaw vitrified samples. And our purpose is to develop cryopreservation protocols for different uses, different type of cells, and teach people how to use them. We've been recently working a lot on mollusks, but also sea urchins and other type of marine cells. We have 3 culture collections within the team of Cryomar. One is Roscoff culture collection. They've been doing cryopreservation for quite a long time. They have control freezers, and they have both storage of liquid nitrogen and storage on ultra freezers, which are electric. And they have at least 31% of their strains in a cryopreserved manner, which I think it helps a lot to maintain, because they do have an extensive culture collection of microalgae and bacteria. Same goes for the Marine Biological Association, MBA, also in the, this is in the UK. And they have facilities dedicated to cryopreservation, freezers. They have both storage of liquid nitrogen and ultra freezers. And they have approximately 76% of their culture in a cryopreserved manner. Okay, and this is quite remarkable. Sam's culture collection, they have a huge amount of strains, around 3,000 strains, and 44% of them are frozen. They have several types of control freezers. They have a liquid nitrogen generator, which is quite nice. And of course, they have cryo storage in liquid nitrogen. And last but not least, from the Netherlands, from the Royal Netherlands Institute of Sea Research, Henk Bulhooy's lab, they've been using cryopreservation, in this case for something very specific. They wanted to preserve natural microbial mats, and I will, this is some of the results they got, simply by using a very basic technique as Mr. Frosty, which actually led them to really good results. So in general, with things we've been doing have several outcomes. We have a couple of chapters on a review book about cryopreservation. Several manuscripts, as you see, have been published, and we published the Cryomark protocol toolbox, which is what I'm going to be showing you right now. This is an assemble of different protocols in a very clear way, step by step, both from the quality of the gametes or cells before freezing to how to do the cryopreservation and thawing, and how to assess the quality of the cells afterwards. So as you can see, things from mussels to oysters to sea urchins to sea cucumbers, many different types of microalgae. There is also macroalgae and fish on top of the microbial mats. So this is how the protocols look like. You can see the kind of materials you need, the regions you need, step by step, everything you need to know to be able to carry them out in your lab. From different species. So what you were seeing before, this is algae, macro and microalgae, but also marine invertebrates, both larvae and sperm and all kinds of cells, and fish. Not only that, we also have another deliverable that was published a little bit ago, where we discuss the technical background on cryopreservation, what is the equipment, the state of the art of the equipment there is available and on the literature right now. And that is also, both these things can be downloaded from the Assemble Plus website, and they're free for anybody to just download and use. And that's it. That's what I wanted to show you. I will be sharing the links through the chat so you can have them. Please use it. I'm pretty sure all the people that it's on those protocols will be happy to answer questions if you have them. But the main idea is they're there publicly to be used by anybody. So please do. And that's it on my side. Before we start on the roundtable, I want to let you all know that we also have a couple of attendees from companies that are technology providers and technology developers. And I will be sharing some of their contacts through the chat so you can actually reach them. I also want to remind you that we have 2 sessions where you can do this one face-to-face meetings. So if you're interested, just get in touch with them, request a meeting, let them tell you everything about the technology they're using. So let me share them with you, and we'll start the roundtable in a second. Okay. Okay. I don't know why my computer is frozen. Can you hear me? Okay. Okay, here we go. This is one of the companies that actually send us some information for you to share. And the other one is right here. And I want to thank you all for joining the session, and please just get in touch with them, see what they're doing. They develop technology in general. They also cater to your needs. And on top of that, I think it's very interesting. They develop a medium for sperm cryopreservation, for fish cryopreservation, and they're probably interested in getting in touch with people that would be happy to test it. So reach out to them. It would be useful for everybody. I'm pretty sure of it.
Estefania, can I jump in and just say that
please.
I sent
this is Christine Campbell from SAMS.
Liquid Night. Yeah, I just want to say that Asinta, the company who supplied our liquid nitrogen generator, because we're about 100 miles from the nearest liquid nitrogen store.
Sorry.
Okay, this secured our supply. So we've had it for 4 years now, I think. And it's never let us down completely, but we have had to get the engineer up a few times, but it has worked very well and saved us money overall. So that's all I wanted to say. Thank you.
Yeah, it's nice to see the point of view of somebody that is actually using this technology. So thank you, Christine. Okay, and then I send you some of the information from the other company. So one of them is a representative from IMV Technologies, and the other one is from Ascent, which is the people that's providing this liquid nitrogen generators. So I think this information could be useful for most of you. So let's just jump into the round table. We will have Niels and Sophia and Juan and myself. And I will just suggest that you ask for a turn and we can just turn on our microphones and talk. Just please not on top of each other, but that will be fine. There's not many of us, and I'm pretty sure we can do it in a, orderly manner. So let me just break the ice. I'm very curious about that super big freezer that Niels showed us into his presentation. It was this design for the company, or you had the idea from some other?
Yeah, I could say that we use a controlled rate freezer, small scale one, planar 360. Then we designed a freezer using a company in Norway called Wins.
Okay.
So they designed this freezer with, which is programmable, and yeah, it have also for the holding periods. It's very stable, because if you have a compressor, you have peaks going up and down like this. But this gives warm air inside the freezer. So then you will have a flat holding period. If you have holding temperatures of -8 or so, you could have it very, very stable. So fluctuates very little. So that's specially designed. That's
well, it looked like, because it was so big, it was kind of out of the ordinary.
yeah.
So that, yeah. I think we had a question for you before. If Damian is still online, Damian, can you turn on your microphone? Well, maybe not. Well, he wanted to know, as for the arguments of using microplankton, you have said that increases growth up to 100%. In which species have you tested this? That's the question he wrote in the chat.
Yeah, so it's a very different response on the growth rates. So in a trial on Ballanrest, we had 100% bigger fish. That was double in size after 3 months or so. And for sea bass, we had 75% higher growth rate on day 40, 50 after hatching. Length was only 10% longer, but they had much more inside, more muscle weight. The myotome height was much larger. Okay. So in length, it's not necessarily so much difference, but in weight, it's often could be a big difference. But it's very different from species to species. And for instance, lumpfish, it could be the same growth the first 30 days, but after 3-4 months, they could be 30-40% larger in size. So it could be a late effect. And also on kingfish, it seems to be a late effect on growth. that is very positive. But
Good. I think that answered the question Damian had.
Yeah.
How about, I think Elsa has a question. Please, just turn on your microphone.
Okay, can you hear me?
Yeah.
Yeah, okay. Nice presentation, Niels, and very, very interesting idea. I just had some doubts, because I think you said that you freeze the cysts, right? The eggs, and well, the eggs, and then they hatch during the thawing process. That's what I understood. So is it they hatch because it's a physical stress, or they hatch naturally because they are under conditions for hatching?
Yeah, so you could say that those eggs, they are actually not eggs. It's a fully developed nucleus inside egg shell, but they are immature when we freeze them, and we are not able to hatch many of those without freezing them. But it seems like this eggshell becomes bristle or something, so it breaks during this freezing process. So after thawing, you see they are popping up like popcorn when you thaw them. You see in the microscope they are popping. So that's very interesting.
And the nauplii, they maintain alive in the tank, right? So they swim, so you can maintain them alive for the fish larvae to prey on them.
Yeah, they swim, but we have not been able to grow them further. So we have been given a different type of algae diets, high quality algae, but have not managed to get them more than stage 2, nauplii stage 2. So we think that when we take them out, when they are immature, they have not the trigger that would bring them to further molting stages. That's my idea, but we are not sure about this.
Okay, thank you very much.
I think we have some question from Hank. Can you please turn on your microphone?
Yes. Hello. Thank you. Thank you very much for these nice presentations. It's very, very interesting. I have a question actually directed to both I forgot the name, and Juan, and the one from the Nautilus. No, what was it? I forgot your names. Niels, sorry. Yeah. Did anybody of you ever do a kind of genomic analysis? And I guess that's very difficult for a blue shark, but of your organisms before and after cryopreservation. Is there any genetic changes occurring due to this process? It may be not so interesting for when you use it directly as food, but it may be interesting if you would return species back into the wild. I mean, I know that in some cases, bacteria and other organisms, they may have some mutations when they are being stored at lower temperature for a while. So I just was wondering whether something like that is checked or possible to check in your systems.
We have not done anything like that. So in-depth analysis like that, we have not.
Yeah, maybe it's out of the box, but I was just, this was one of the things that popped up in my mind. Like,
but a very interesting question.
No, the question is very interesting. In the case of sharks and rays, we are at the very beginning. For the moment, we know that after thawing, a good percentage of the spermatozoa are able to swim. They have motility capacity, and the membranes apparently are quite good, quite well. We are not damaging the membranes. But as you ask, we have no idea what is happening with the DNA. Probably the DNA is having some damages that we should evaluate, but for the moment we have not arrived at this point.
Okay. Gracias. Tak.
You're welcome, Hans. So I'm not sure if, I think Elsa had an opinion on that.
Sorry, it's just from this question from Juan. Yeah, I think it's quite interesting that you could do some further analysis, because since it's for conservation of species, so I think it's very important. I was just wondering if you plan to do, or you just said that you are in the early beginning, and of course you are working with very complicated species and with a lot of species, so it's difficult. But do you plan to do any artificial fertilization, or if you plan to do it, how it would be in this case of the sharks, because they are very different from the fish that we usually work.
Yes, we are talking about species with internal fertilization,
Yeah.
and this is something different. And we are talking about fish that produce the envelope of the egg after the fertilization of the egg. So this is giving us new challenges, new things to consider, and well, this is at the same time a problem and a good challenge to think about. Our intention is thinking in at least trying to develop these techniques, having in mind the use in conservation programs for endangered species, and at the same time considering the possibility of applying these techniques in public aquaria over the world, because they have species in captivity, and many times they have the male or the female, but not both together. And if they want to think in reproduction, they must think in sending one of the members of the couple, the male or the female, in a plane or a bus or a truck or whatever. And this is difficult, this is expensive, and sending a sample of frozen sperm, it will help a lot this company.
Yeah, it's true. Thank you.
Thanks to you.
Yeah, I would just wanted to briefly say to change subject, but before, Sophia and their team from the University of Vigo, they're from the genetic lab, and they're experts on this kind of thing. And we were kind of wondering if we could test what happened with the genetic composition of the larvae after we, cryopreserve it and store it, to make sure we're just kind of storing the same thing and bringing back exactly what we're looking for. Because if you're going to biobank a species because their genetics is valuable, you don't want to mess up with that. So we wanted to have a look and check how that would turn out. But we're still on our very first stages.
And we were talking about that with Angel some days ago, and we were thinking, okay, we know how to do that proteomic analysis, for example, or genomic analysis of those samples or larvae or sperm after the reactivation, for example. And before we were thinking, we don't know if it's better to just, for example, with a sperm, it's not the case, but to take it like fresh and try to conserve it, for example, with a buffer. We use a lysis buffer to denaturate the proteins, to take them. And or if it's better to do the first analysis, like how is the proteome before the cryopreservation, first to cryopreserve in nitrogen, in liquid nitrogen, and then do the analysis. Because we was thinking, if we spend too much time to do the analysis, maybe the proteome or the genome of even for example, the sperm that is moving all the time fresh is changing. So we don't know if it's better to do it fresh or cryopreserve and do the analysis right there. And later, yes, okay, reactivate and do it later. I don't know if I explained myself.
Well, I'm not sure what is the right answer right now. But I would probably think that with larvae is not that crucial, because they're not, okay. It's not like a sperm that they have such a short shelf life and they're spending their energy by the second. Larvae is not that active, and I think we should be fine to do it before and right after cryopreservation.
Yes, because I was thinking maybe the cryopreservation, when you just get into the nitrogen, maybe there are being changes than I don't know.
But you can separate.
In contact
I don't think we can separate both processes. So
no.
We'll see.
Okay, yes.
I had a question for Juan. So you were showing a picture of how different the sperm from sharks look than other animals that you've worked with. How about eel? Is eel sperm too different from fish?
Well, in fish we have a huge diversity in terms of morphology. Eels has the head of the spermatozoa of the eels is like a half moon, more or less, has a shaped curve with a mitochondria in the apical part instead of being near the flagellum linking or near the link with the flagellum, and well, a part of this is quite regular in terms of size and this kind of aspects. The lasmorans are different. The lasmoran has this. In the case of sharks, they are like a screw. There's a straight line, straight structure with this corpse around. And in the case of rays, is I don't know the English word. How do you call this tool to open wine bottles? Having this circular structure. Well, you understand me.
I don't know.
When you screw one
screw opener, I'm not sure.
This structure with curvature is like open. It's different than the structure of one screw that is the shark one. And we are seeing that the number of cores, for instance, is species specific, in both in rays and sharks. And well, we are another characteristic of this sperm is that during the fertilization, the spermatozoa are not free. They are linked in masses, in groups called spermatozomatas, and only when the female is ready for the fertilization, when the female decides to use the sperm, then these groups are dissolved, and then the isolated cells have the capacity of separate and start swimming.
So I'm guessing you can't freeze them when they're like all together.
Yeah. This could be a possibility. This could help or not. Maybe having a free cell where the cryoprotectants could arrive in all the surface could be better. So we have this doubt, this question at this moment.
So we're just reaching the end. Thank you for coming. Just a final quick question for all the participants. Nils, what is the main constraint you found on using cryopreservation on planktonic?
The main constraint for using cryopreservation.
I don't know if you overcome the constraint or it's still there, but what was the main thing you have to fight against?
This was scaling up. I think that was a major problem, to scale up from vials to larger volumes. So that had to be taken step by step, several years. So and when you solve one bottleneck, you find the next one. So it has been challenging, but especially the controlled rate freezer, I would say, have been a challenge, especially with our protocols, because they don't fit into to a normal controlled rate freezer, because we are doing a long time freezing. That's the only thing that works on our organism and the protocol we are using. So that have been the main challenge, I think.
Okay, but it seems you had it under control, both of them, so that gave us everybody else hope with their bottlenecks.
Yeah.
Well, Sophia, I think the main constraint you already told us is to make sure genetics is still preserved. We can hear you. You need to turn on your microphone.
Sorry. Okay. Yes. Also because working, I think, with every animal, with something that is alive, they are the time, okay, we obtain the larva, and there's a lot of larva, and try to work with them all of together and try they not grow too much once and the other not, try to all of them get uniform. And of course, we are in the genetic group working with proteomics. So yes, we always want to know what's happening before and after, or what changed, because those changes could be involved later in the phenotype. We are interested on those lines. So it's important to preserve that. So yes.
Thank you. How about you, Juan? What is the main constraint?
Well, apart of the money, that I suppose this is something common, it's the lack of knowledge. We are, at least in the case of elasmobranchs, we are in a totally unexplored area. So this is a problem, because you cannot compare your results with nothing, basically. But at the same time, it's very exciting. So, well, let's go.
Awesome. Well, this is the end. We need to move on to the B2B session. So feel free to contact somebody else if you're interested in starting a venture, project together, or whatever. Please use the platform, get in touch with each other. It's going to be available for a couple more days, so you have time. And just, well, I want to thank you all. I think it was very interesting to see the perspective from a company that's already on the run, from a project that may or may not have a spin-off and turn into a venture, and also from Juan, who's saving biodiversity for our next generations. So I hope it was very useful, and thank you for being here.
Thank you.
Thank you.
Okay, bye.
Bye.
Bye. Thank you.
The marine environment is a rich and largely unexplored reservoir of biodiversity with vast potential for food, health and biotechnology. The European Marine Biological Resource Centre, EMBRC, is a research infrastructure which aims to unlock the knowledge and innovation potential of our oceans. It enables researchers and companies to access marine organisms, expertise and experimental facilities to study them. Headquartered in Paris, EMBRC brings together 45 sites in 9 member countries.
We provide access to specialist facilities and services that enable researchers from academia and industry to study marine life and develop innovative solutions to address societal challenges like climate change and health and food sustainability. We support both fundamental and applied research, particularly for areas like biodiscovery, biotechnology, aquaculture, biodiversity, and climate change research. EMBRC-supported research has already led to novel, high-impact research in human health, product and medicine development, and aquaculture, and it's helping us to fully grasp the crucial role of ocean life.
EMBRC has benefited hundreds of researchers across Europe and beyond, delivering robust and efficient services and expertise to help users obtain the best possible results.
So EMBRC is continuing to develop its services. We're working to start recording biodiversity at many of our sites using molecular techniques to put in place so-called genomics observatories. This will allow us to have a much better understanding of how our oceans function and their current health. In addition, we're increasing our bioprospecting capabilities to better support the development of new products and solutions from the sea.
EMBRC is a single access point to remote and on-site services in Europe, supporting marine research and innovation across borders.